Have you ever really thought about how scientists actually rewrite the code of life? I mean, it's not like they're just typing on a keyboard. It's way more like a high-stakes, super technical heist. The target? A fortress so small you can't even see it. And the prize? Well, the prize is the power to change biology itself. So let's gear up and break down exactly how this cellular heist goes down.
Right, let's frame it like this, something we can all get our heads around. The big question is, how in the world do you install new software, a piece of DNA, onto the most ridiculously complex computer on the planet, a living cell?
Okay, so take a look at this bacterium. Every single cell is basically a fortress. It's wrapped in this incredibly sophisticated membrane that is just fantastic at its job, which is keeping foreign stuff out. And you guessed it, that includes the very DNA we're trying to sneak inside. But here's the cool part. Over the years, scientists have gotten really good at this. They've developed this amazing toolkit of techniques. They've become master locksmiths, getaway drivers, and sometimes they just know how to blow the doors clean off. All right, first things first. Before any heist, you gotta case the joint. We need to get a really good look at the defenses of this cellular fortress to have any chance of getting inside. So here it is, the main security system. Big molecules like DNA just can't cross the cell's membranes by themselves. No pass, no entry. The fortress is sealed tight. If we want to get our genetic package across these walls, we're going to need a clever plan, a skeleton key, or, a battering ram. Let's start with the subtle approach. The first tools in our kit are what I like to call the chemical keys and disguises. These are all about tricking the cell into lowering its guard and just letting our package right in. So step one in this whole con job is to make the cell competent. What does that mean? Well, it means we treat it with some specific chemicals that, let's say they rough up the security guards a little bit. They weaken the walls, making the cell vulnerable and primed for a break-in. And this right here is the classic move, the heat shock. It's brilliant. First, you chill these competent cells way down in an ice-cold calcium chloride bath. This gets the DNA to just kind of stick to the outside of the cell. Then, bam, a quick, intense blast of heat. We're talking 42 degrees Celsius, and it startles the cell, forcing it to gulp down whatever DNA is sitting at the door. It's a fantastic 1-2 punch. And there are other super clever chemical tricks, too. On the left, you've got this diversion tactic. Scientists use calcium phosphate, which makes the DNA clump up and stick to the cell surface, where it kind of gets swallowed by accident. Or check out the image on the right, the perfect disguise. It's called a liposome. It's a tiny bubble made of the exact same fatty stuff as the cell membrane. You just hide the DNA inside, and the bubble merges right with the cell, delivering its cargo like a secret agent. But, being subtle doesn't always cut it. Sometimes you've got to forget the lockpicks and just go in loud. It's time to talk about brute force. Now, the brute force toolkit has 3 main instruments of persuasion, each one more wild than the last. We've got the zap, we've got the needle, and my personal favorite, the cannon. First up, the zap, also known as electroporation. For this trick, you put your cells in a little bath with the DNA, and then you hit them with a high-voltage electric pulse. This jolt literally zaps tiny temporary holes into the cell membrane. For just a split second, the gates are wide open, and the DNA can just slip right on through before the wall seals itself back up. And listen, this isn't just some flashy trick. The zap is incredibly effective. We're talking 10 to 20 times more effective than your standard chemical methods. When you absolutely need to get that code inside, you bring in the electricity. Next up, we have the needle. And yeah, it is exactly what it sounds like, but the scale is just mind-blowing. Microinjection uses a glass needle so fine it's thinner than a human hair, and with it, you physically inject a DNA solution directly into the heart of the cell, the nucleus. I mean, this is basically surgery on a single cell. The precision required is just, wow. And finally, we get to the most action-packed method of the whole bunch, the cannon. The official name is biolistics, but what it is a gene gun. It fires microscopic bullets, tiny particles of gold or tungsten that have been coated with DNA at unbelievable speeds. These little micro-projectiles literally blast their way through the cell wall and membrane to deliver their payload. It's just wild. So you might be asking, why on earth would you use such an aggressive tactic? Well, the huge advantage is that you can target very specific tissues inside a living organism, not just cells floating in a dish. Of course, the downside is pretty obvious, right? There's a real risk of collateral damage. I mean, you are firing projectiles at microscopic targets. So, we've seen the sneaky chemical tricks and the loud brute force methods. But maybe the most elegant solution of all is to just stop fighting the system and instead use nature's own master infiltrators. It's time to bring in the Trojan horses. The strategy here is just beautiful in its simplicity. Think about it. Instead of trying to fight your way past the cell's defenses, why not use an agent that has spent millions of years evolving to do exactly that? I'm talking, of course, about viruses. So what scientists do is this amazing thing called in vitro packaging. In a test tube, they mix their custom-made DNA with the protein parts of a virus. And then something incredible happens. The parts just self-assemble. They build themselves into complete infectious viral particles. But the key is, the dangerous viral genes have been swapped out for our new code. The Trojan horse literally builds itself, ready to deliver our message. And for plants, it turns out there's another natural master hacker out there. It's a bacterium called agrobacterium. This little organism evolved to do the exact things scientists want to do, inject a piece of its DNA into a plant cell. So scientists just hijack this natural system, swap out the bacterium's DNA with their own, and let it do all the hard work for them. Okay, so we've got chemical persuasion, we've got brute force, we've got biological Trojan horses. But why? Why go through all this trouble? What's the ultimate payoff for pulling off this ridiculously complex cellular heist? This is it. Mission accomplished. Every single one of those glowing green colonies you see on this plate, that's a group of bacteria where our heist was a success. They took in the new DNA, and now they're expressing it by making a fluorescent protein. It's the beautiful, visual proof that we did it. We successfully rewrote their code. And the stakes here could not be higher. Every single one of these world-changing advances, gene therapies that can cure inherited diseases, crops that can survive a drought, bacteria that can produce life-saving insulin for us, even cutting-edge DNA vaccines, every single one is built on the success of this cellular heist. This is the absolute bedrock of modern biotechnology. You see, we're not just readers of the genetic code anymore, we're becoming its authors. The ability to pull off the cellular heist has given us the power to edit life itself. And that leaves us with the biggest, most important question of all. What story should we write next?